If you were to look at an MRI scan of an adult patient, maybe someone struggling with severe schizophrenia,
Yeah.
you might notice some subtle structural changes in the brain.
Right, like a slight enlargement of the ventricles, perhaps.
Exactly. Or maybe a thinning of the cortical gray matter. And usually when we talk about a medical diagnosis, there is this sort of expectation of precision.
Yeah, you break your arm, the x-ray shows a jagged white line, and that's it.
Spot on. The doctor points at it and says, there it is, broken.
Yeah.
We like things to be visible, to be neatly categorized, and to have a clear beginning and end.
It's comforting.
It is. But what if I told you that the blueprints for the structural changes you see on that adult MRI were drawn up, and the fatal error was actually made weeks before that patient was even born?
It completely upends our standard medical intuition, doesn't it? Because in psychiatry, we're rarely looking at a sudden acute break.
Right.
We're looking at the culmination of a developmental trajectory that spans decades. A trajectory tracing all the way back to the very first trimester of gestation.
And that is exactly where we're taking you today in this deep dive. Because at its core, psychiatry is really the study of how brain, mind, and experience interact.
And crucially, how that intricate process can go wrong.
Absolutely. I mean, if our genomics, our DNA, provides the raw script for who we are, then neural development is the unfolding, live performance. It's the stage where the actors actually have to hit their marks.
That's a great way to frame it.
Thanks. I always used to imagine that the brain was constructed statically, like a building. You lay the foundation, you put up the walls according to a rigid blueprint, and then you're just finished.
Right. But looking closely at the developmental neuroscience, it's clear that analogy falls apart entirely.
It really does.
A much better way to conceptualize it is to think of the brain as a masterpiece being actively sculpted from a massive block of marble.
I like that.
And the critical insight here is that when you sculpt marble, what you physically strike away and remove is just as important as the stone you initially leave behind.
So the negative space matters just as much.
Precisely. In neurodevelopment, what is aggressively pruned away over time is just as vital as what is built.
Or, to push that even further, imagine a dense, overgrown jungle. To make it navigable, you have to cut back the vegetation to form a specific trail system, right?
Yes, exactly.
If you don't cut back the excess vines, you just get lost in the noise and sensory overload. But if you slash and burn too much, there's no trail left to follow at all.
That is a brilliant metaphor for what we're going to cover.
Today, we are going to explore how the brain grows, shapes itself, and learns exactly what to keep. We'll trace this journey from a simple, microscopic flat sheet of cells in a tiny embryo all the way up to the most complex structure in the known universe.
It's quite a journey, and we want to build mental models so you can understand why mature psychiatric conditions or developmental conditions like autism are deeply rooted in these early microscopic cellular dances.
So to build those models, I suppose we need to establish the foundational rules of construction first.
Right. We do. We need the rules that govern how this organ puts itself together. And the very first principle we have to grasp is that timing creates windows of susceptibility.
Because the brain isn't just built all at once on a Tuesday.
No. Different regions and different populations of neurons are generated at distinct, highly specific times.
So if a fetus is exposed to a toxic insult, say, a severe maternal infection or some sort of teratogenic drug at week 4 of pregnancy, it's going to paint a completely different clinical picture than if that exact same exposure happened at week 24.
Vastly different. An insult at week 4 might disrupt the entire central axis of the nervous system. It could potentially prevent the brain from forming at all.
Wow.
But that exact same insult at week 24 might only affect the fine-tuning of the outermost layers of the cerebral cortex, or perhaps the development of the cerebellum. The temporal window dictates the vulnerability. Time really is the canvas here.
Which flows naturally into the second rule, which is the cascade effect. Development is sequential. It's this ontogenetic domino effect.
Spot on. If you have an abnormality in a very early cellular event, it will inevitably lead to deviations in every single subsequent stage that relies upon it.
But the tricky part for clinicians is that those early primary abnormalities are often completely invisible to our current diagnostic tools, right?
They are. They're hidden beneath years of compensatory changes.
This actually reminds me of pouring the concrete foundation for a house. If the contractors pour the foundation just slightly off level, say, by just a few millimeters, nobody notices at first.
Right, the walls go up fine.
Exactly. The framing looks fine, the drywall is painted, but 10 years later, because the weight distribution has been subtly wrong the entire time, the roof starts leaking.
That is the perfect clinical translation.
So a patient might come into your clinic with adult symptoms, severe anxiety, psychosis, profound depression. We are essentially treating the leaky roof. But the true origin, the root cause, was a microscopic foundation error made decades earlier.
Yes, and you might order an MRI on a 25-year-old patient and see a reduction in axonal white matter. But that reduction might actually be the downstream consequence of a deficit in the sheer number of early-born neurons that failed to generate back in the first trimester.
Because the neurons weren't there, they couldn't project their axons.
Exactly, hence the missing white matter. We are diagnosing the late consequence while the initial error remains completely hidden in the past.
It really shifts how you view a psychiatric presentation. Now, the third rule of construction seems to be an exercise in extreme biological efficiency. The brain reuses its molecular signals.
It does. Evolution is incredibly conservative with its tools. The extracellular growth factors and transcription factors that the brain uses are deployed repeatedly.
So it recycles the same chemical instructions for totally different jobs.
Precisely. In the early embryo, a specific chemical signal might simply tell a group of precursor cells to multiply.
Okay.
A few months later, that exact same chemical signal is deployed to tell newborn neurons where to migrate. And years later, in the adult brain, that identical signal is suddenly used to drive synaptic plasticity.
Meaning the microscopic structural changes that actually allow you to form a new memory.
Yes.
The implication of that is staggering, though. If a patient carries a genetic variant that slightly alters one of these chemical signals, it isn't just going to break one specific event in their life.
No, it's going to ripple through their entire timeline. It might slightly alter their cell proliferation early on, then subtly shift their neuronal migration, and later in life impact their cognitive flexibility.
Which neatly explains why modern psychiatric genetics is so incredibly complex. We constantly see the same risk genes implicated across multiple, seemingly distinct clinical diagnoses.
Like schizophrenia, bipolar disorder, and autism. It is precisely because the underlying molecular tools are shared across the entire lifespan.
Right. And that brings us to the fourth rule, which focuses on our unique human trajectory. We have an exceptionally high capacity for neurogenesis.
We do. And we maintain a prolonged period of maturation compared to almost any other species.
Our brains just take an incredibly long time to finish baking.
They do, and this extended maturation is what grants us our profound capacity for extended learning, social complexity, and memory. But it's a double-edged sword.
How so?
Well, an extended maturation period intrinsically means an extended window of vulnerability. Our neural circuits remain susceptible to environmental insults, stress, and trauma for much, much longer than a rodent or even a non-human primate.
So we have our rules. Timing dictates vulnerability, errors cascade, signals are recycled, and our maturation is dangerously prolonged.
Exactly.
With that framework in mind, I want to take you back to the very beginning, to the embryo between weeks 2 and 4 of gestation. We're looking at the transition from gastrulation to neurulation.
Right. At around day 13, the human embryo is basically just a flat sheet of cells.
Which is hard to even picture as a human.
It is. Following gastrulation around days 14 and 15, this embryo organizes into a two-layered sheet. You have the ectoderm on the outside and the endoderm on the inside.
But to build a human, and specifically a nervous system, we need a third layer.
Yes, so cells migrate into a midline cleft in the ectoderm, which is known as the primitive streak, to form a middle layer called the mesoderm.
And this newly formed mesoderm is where the magic of induction begins, isn't it?
It is.
I found this process absolutely fascinating when reading the chapter. The underlying mesoderm, specifically a rod-like structure called the notochord, starts releasing a chemical soup of soluble growth factors.
Correct.
But how on earth does a chemical soup tell a completely generic, blank slate cell sitting above it to become a highly specialized brain cell?
Well, it comes down to receptors and transcription factors.
Yeah.
You can think of transcription factors as molecular switchboard operators inside the cell's nuclei.
Okay, switchboard operators.
The growth factors released by the notochord float upward and bind to specific receptors on the surface of the ectoderm cells. This physical binding triggers a signal that travels down into the nucleus, activating these switchboard operators.
So they get the call, so to speak.
Exactly. They physically bind to the cell's DNA and start turning specific genes on and off. The chemical soup from the mesoderm is literally rewriting the genetic destiny of the ectoderm above it.
But the twist here is what that default destiny actually is. You would assume that the default state of the outer layer, the ectoderm, would be to become skin.
That would be the logical assumption.
Right. And that it requires a special complex instruction to become a brain. But it's the exact opposite.
It is deeply counterintuitive. Left to its own devices, the ectoderm naturally wants to become neural tissue.
Wait, really?
Yes. The only reason it becomes skin is because neighboring cells are actively secreting molecules called bone morphogenetic proteins, or BMPs. These BMPs are signals that scream, turn into epidermis, become skin.
Wow. So to build a brain, the notochord doesn't actually tell the cells to become a brain at all.
No, it just blocks the skin-forming signal.
It secretes inhibitors of BMP. It's essentially stepping in and shouting, ignore them. Stay neural.
That's exactly it. The notochord provides spatial boundaries by blocking the epidermal fate. And this delineates the neural plate.
And once this plate is established, around day 18, it's just a flat, thickened sheet of cells.
Right, and then the lateral edges of this plate start to elevate, forming neural folds or ridges. Driven by physical changes in the cell's internal cytoskeletons, these ridges fold inward, meet in the midline, and zip up.
And that zipping process forms the neural tube, the fundamental architecture of the entire central nervous system.
Exactly. And the hollow cavity trapped inside that tube will eventually become the brain's ventricular system, filled with cerebrospinal fluid.
And this zipping up, or neurulation, doesn't happen all at once, does it?
No, it starts in the middle, in the cervical region that will eventually become the brainstem, and it zips in both directions, upwards towards the head and downwards towards the tail.
But clinically, we know the zipper can get stuck.
And when it does, the clinical manifestations are devastating. If the neural tube fails to close rostrally at the top end, it results in anencephaly.
Which is where the major portions of the brain and skull entirely fail to develop.
Yes. And if it fails to close caudally at the bottom end, the result is spina bifida, where the spinal cord remains exposed to the amniotic fluid.
This is a scenario where embryology directly intersects with global public health. We know that certain environmental factors and medications severely disrupt this delicate folding process.
Absolutely. Take valproic acid, for instance.
Right, a very common anticonvulsant frequently used in psychiatry as a mood stabilizer for bipolar disorder.
Yes, and if a patient takes valproic acid during those crucial early weeks of pregnancy, it drastically increases the risk of these neural tube defects.
Which is why prescribing guidelines for women of childbearing age are so incredibly strict regarding valpro.
Precisely. Furthermore, we know that a maternal diet deficient in folic acid impairs the cellular machinery required for the tube to fuse.
Which explains why folic acid supplementation is a global mandate. We are intervening directly in that early chemical soup to ensure the zipper has the metabolic support it needs to close.
It's quite remarkable.
It's incredible that a simple vitamin supplement can prevent a catastrophic structural failure of the nervous system. Now, as the neural tube is zipping up, there's a distinct population of cells right at the crest of the fold that absolutely refuses to be locked inside.
Ah, the neural crest cells. As the tube merges, these cells pinch off from the dorsal aspect, the top of the closing tube.
And they undergo an extraordinary migration.
They are the great wanderers of the embryo.
Some of them migrate ventromedially, deep into the body, to form the peripheral nervous system. They form the sensory ganglia along the spine, and they travel all the way down into the gut to build the enteric nervous system.
But others take a completely different path. They migrate dorsolaterally, spreading out just underneath the developing skin.
To become melanocytes, the pigment-producing cells of our skin.
Yes.
When I realized this, it was a massive light bulb moment for me. Because if you spend time on psychiatric or neurological wards, you will inevitably encounter patients with neurocutaneous disorders.
Conditions like tuberous sclerosis or neurofibromatosis.
Exactly. These conditions bridge specialties. And they always seem so perplexing to me. A patient will present with profound neurological and psychiatric symptoms, intractable epilepsy, cognitive differences, autism spectrum presentations.
Right.
But their diagnostic criteria also hinge on very specific, distinct skin lesions or pigment changes. You look at it and wonder, what on earth does a patch of discolored skin have to do with severe seizures and neurodevelopmental delays?
And embryology provides the definitive answer. The affected brain circuits and the affected skin pigment cells share the exact same embryonic origin.
They were both born from that specific population of neural crest cells that pinched off at week 3 of gestation.
Exactly. If there is a genetic mutation in that ancestral crest cell, it will inevitably manifest in both the central nervous system and the skin.
The skin is literally a visible dermatological window into the brain's invisible developmental history.
It really highlights how artificial our medical specialties can be sometimes. The embryo certainly doesn't know the difference between dermatology and psychiatry.
It's so true. Okay, so we have a fully closed neural tube, but right now it's just a simple pipe. To build a brain, this pipe needs to expand and partition into specific rooms.
At 4 weeks of gestation, the head-to-tail axis of the tube undergoes differential expansion. It divides into 3 primary vesicles.
The prosencephalon, which will become the forebrain, the mesencephalon, the midbrain, and the rhombencephalon, the hindbrain.
Correct. And by week 5, those 3 rooms subdivide further into 5. The prosencephalon splits into the telencephalon, which becomes the heavy hitters like the cerebral cortex, the hippocampus, the basal ganglia.
And the diencephalon, which forms the thalamus and hypothalamus.
Right. But here is the major conceptual hurdle. As these massive expansions are happening, the cells lining the inside of the tube all look completely identical under a microscope.
So how do they know what region they're supposed to build? How does a generic precursor cell know it's supposed to generate the motor cortex instead of the visual cortex?
This introduces one of the most elegant concepts in neurobiology, the protomap.
The progomap.
While the cells in the ventricular zone, which is the innermost lining of the tube, might look identical physically, their genetic expression is vastly different. They express a checkerboard array of transcription factors.
Okay, so a genetic checkerboard.
Yes. These factors are distributed in overlapping gradients across the ventricular surface, effectively drawing a map of future brain regions long before those regions actually exist.
Let's ground this with a specific example for the listeners. There are 2 heavily studied patterning genes in the forebrain called PAX6 and MX2.
Yes. The anterior or front part of the developing cortex, which is destined to become the motor cortex, expresses very high levels of the transcription factor PAX6.
Meanwhile, the posterior part, destined to become the visual and sensory cortex, expresses high levels of MX2.
Exactly. And they don't just passively occupy their space, they are highly territorial. They actively push against each other.
So PAX6 structurally suppresses the expression of MX2, and MX2 suppresses PAX6. By mutually inhibiting each other, they establish a hard physical boundary line.
They are staking their claims on the cortical real estate.
Right. So let's play this out clinically. What happens if a genetic mutation deletes PAX6 entirely? Does the embryo just develop a brain with a giant empty gap in the front where the motor cortex should be?
Not an empty gap, actually. Because of that reciprocal inhibition, if you remove PAX6 from the equation, MX2 is no longer being suppressed.
Oh, I see.
The sensory territory, driven by MX2, aggressively expands forward into the void. The boundary line shifts entirely.
Wow. So you end up with a brain that has a massive overrepresentation of sensory cortex and a drastically shrunken hypoplastic motor cortex.
Precisely. The physical map of the brain's functions is completely rewritten just because a single molecular boundary fence was removed.
A single gene shifts the entire geography of the mind. And we see the consequences of this in human patients, don't we?
We do. Mutations in PX6 cause profound malformations. You don't just get a minor motor deficit, you get a severely underdeveloped cerebral cortex.
And because of the cascade effect we talked about earlier, it also disrupts eye development, causing aniridia, the absence of the iris, and poorly formed olfactory bulbs. One patterning error ripples through multiple sensory and motor systems.
It's all connected.
So the protomap dictates what the regions will become, but where are all these billions of brain cells actually being manufactured?
The primary factory floor is the ventricular zone, or VZ. This is the densely packed layer of precursor cells that lines the walls of the entire ventricular system.
Early on, it's just a single cell layer thick, right? A pseudo-stratified neuroepithelium.
Yes, and this factory floor features a truly bizarre physical phenomenon called interkinetic nuclear migration.
This part is wild. The precursor cells are these elongated bipolar structures. One end touches the ventricular space, and the other stretches outward toward the developing surface of the brain.
Right. But their nuclei, the bulky compartment containing the DNA, act like elevators, constantly moving up and down the shaft of the cell.
It is a mesmerizing process to visualize. The cell's replication cycle is physically mapped to the location of its nucleus.
Yes. When the cell needs to replicate its DNA, which is the S phase of the cell cycle, the nucleus rides the elevator up, moving away from the ventricular surface.
And once the DNA is fully doubled, the nucleus rides the elevator back down.
And when it hits the ground floor, the surface of the ventricle, that is where it finally undergoes mitosis and divides into 2 cells.
But why go through this elaborate, energy-intensive physical dance just to divide?
Well, there are two prevailing theories. The first involves signaling. Dividing cells possess primary cilia, which are tiny, hair-like sensory antennae that physically project into the ventricular fluid.
Okay.
By dropping down to the surface, the dividing cell can use these cilia to sense local regulatory signals floating in the fluid, like sonic hedgehog or want proteins.
So they're dipping their toes in the water to get the latest instructions.
Exactly, to know whether to keep dividing or to stop. The second theory is purely mechanical. The ventricular surface is incredibly crowded. By moving their bulky nuclei away to replicate their DNA, the cells are literally making physical room at the surface for the cells that are actively splitting.
It's like students leaving a crowded lecture hall to go study in a quiet library and only coming back down to the crowded room to take the actual exam. It manages the traffic.
That's a lovely way to picture it.
But here's the catch. The ventricular zone factory is efficient, but it isn't nearly enough to build a human brain. I mean, a mouse has a ventricular zone, but its cortex is tiny and perfectly smooth.
Exactly. If evolution had stopped with the ventricular zone, our brains would cap out at a very small number of neurons. The evolutionary leap that allowed for higher mammals, and specifically primates, was the development of secondary proliferative zones.
Right, the subventricular zones.
First, the subventricular zone, or SVZ, evolved to produce intermediate precursor cells. But the true game changer for the human brain was the massive, unprecedented expansion of the outer subventricular zone, the OSVZ.
This is really the secret to our massive cognitive processing power.
In the second trimester of human development, the OSVZ becomes the dominant site of neurogenesis. It churns out neurons almost 4 times faster than the primary zone.
And it achieves this using a remarkable cell type called the outer radial glia-like cell, or ORG.
Yes. Unlike the standard factory workers that are tethered to the ventricle and forced to ride the elevator up and down, these ORG cells have unhitched themselves. They detach from the ventricle and only have a process reaching out toward the outer surface.
Like, because they're detached from the surface, they don't have to waste time doing that interkinetic nuclear migration all the way down to the ventricle.
Exactly. They stay in the outer zone, multiply rapidly, and massively amplify the production of neurons.
And the physical consequence of generating millions of neurons at such an exponential rate is that the developing cortex literally runs out of room inside the confines of the skull.
So to fit all that new surface area into a restricted volume, the cortex is forced to buckle, fold, and collapse in on itself,
Creating the complex gyri and sulci, the hills and valleys that give the human brain its characteristic wrinkled appearance.
We refer to this as a gyrencephalic brain. And what is truly groundbreaking in recent neuroscience is that we're beginning to identify the specific genetic drivers of this evolutionary leap.
Such as?
Researchers have highlighted primate-specific genes, such as NOTCH2NL. These genes appear to specifically turbocharge the proliferation within the OSVZ.
It is breathtaking to consider that a few recently evolved genetic tweaks are directly responsible for the sheer volume of cortical neurons that allow us to process language, develop complex societies, and even understand our own biology.
It really is.
So thanks to this turbocharged factory, we have now mass-produced billions of neurons. But right now, they are all piled up deep in the center of the brain near the ventricles.
A massive pile of cells isn't a functional brain.
No, it's not. They have to travel outward and wire themselves into a highly specific geographic architecture. How do they know where to go?
This is where we transition from proliferation to migration. The classic, most well-studied pattern for building the cerebral cortex is inside to outside radial migration.
Radial meaning they travel in a straight line from the deep ventricular center outward toward the skull. But they're essentially migrating through a dense swamp of tissue. They need a guide.
They do. They utilize specialized cells called radial glial cells as a physical scaffold. Think of these radioglia as a dense system of vines climbing up a trellis.
Or ropes drop straight down from a helicopter.
Yes, exactly. The newborn neurons wrap their bodies around these radial glial fibers and physically crawl their way outward.
But before they start building the permanent 6 layers of the adult cortex, there is a fascinating temporary structure that gets laid down first. It's called the subplate. It functions like a fetal waiting room.
The subplate is a crucial transient layer. Long before the permanent neurons of the upper cortex have fully arrived, and long before the fetus is receiving any meaningful sensory input from the outside world, the neurons in the subplate are electrically active.
Wait, really? They're firing spontaneously.
Yes. It is as if they are testing the cables, practicing the complex wiring of the brain in the dark of the womb.
Oh, wow. In fact, the sensory cables coming up from the thalamus, the thalamic afferents, actually plug into the subplate temporarily, don't they?
They do, waiting for their final cortical targets to be built above them.
And this practice wiring makes the subplate incredibly important from a clinical perspective. These highly active neurons are profoundly vulnerable to perinatal insults.
Yes. If a baby is born prematurely and requires care in the neonatal intensive care unit, the NICU, they are often at risk for hypoxia or ischemia, a lack of oxygen or blood flow to the brain.
And these insults can selectively devastate the subplate, disrupting that critical early wiring practice.
Exactly. This is believed to be a major underlying mechanism for the lifelong neurodevelopmental consequences, such as cerebral palsy or cognitive delays that we often see in premature infants.
Right, so the subplate is waiting and practicing. Meanwhile, the main cortex is building itself in a highly counterintuitive inside-out fashion.
Yes. You might logically assume that a structure builds from the bottom up, laying the foundation and then stacking on top.
That makes sense.
But in the cortex, the deepest permanent layers, layers V and dias, whose neurons will eventually project massive axons all the way down to the spinal cord and brainstem, are laid down first.
They migrate up the trellis and settle right on top of the subplate.
Then, the next batch of neurons is born at the ventricle. These are destined for the superficial layers, layers 2, 3, and 4, which will handle local processing and connect the two hemispheres of the brain together.
Okay, so how do they get there?
These newly born neurons have to migrate past the already established deep layers to settle on top of them.
It's like arriving late to a concert and having to push your way through a densely packed crowd to get to the front row.
That's a good way to picture it.
But this raises an obvious mechanical question. If they're climbing up this trellis, how do they know when to stop? What prevents them from just climbing right through the top of the brain and into the meninges?
They rely on a precise molecular stop sign. At the very outer surface of the developing brain, in a region called the marginal zone, there's a sparse population of specialized cells known as Cashal-Retzius cells.
And these cells constantly secrete an extracellular glycoprotein called reelin.
Yes, reelin.
It functions exactly like a traffic cop, standing at the top of the trellis, holding up a hand.
Precisely. As the migrating neuron crawls upward, it eventually encounters the reelin gradient. The reelin signal tells the neuron to detach from the radial glial fiber, stop migrating, and align itself into its proper layer.
But what if that traffic cop is missing? What if the gene for reelin is mutated?
Complete architectural chaos. Without reelin, the migrating neurons do not know where or when to stop. They pile up in a disorganized, inverted, and chaotic pattern.
Just a complete architectural failure.
Exactly. The entire inside-out construction methodology fails.
And the clinical manifestation of this failure is a condition called lissencephaly.
Which translates literally to smooth brain. Because the radial migration fails so catastrophically, the cortex never expands outward properly, and those complex folds, the gyri and sulci, never form.
The surface of the brain remains completely smooth, resembling the brain of a much lower mammal.
Yes, and for the patient, the consequences are profound. Lissencephaly results in severe intellectual disability, profound developmental delays, and intractable treatment-resistant epilepsy.
But reelin isn't the only point of failure. The cellular engine driving the migration can also break.
Yes, the neuron physically pulls itself up the glial trellis using an internal engine made of microtubules. If the genes coding for microtubule-associated proteins such as double cortin or DCX and LIS1 are mutated, the engine stalls.
The cell physically cannot migrate, even if the reelin traffic cop is present.
We see this clearly in X-linked lissencephaly. Males with a DCX mutation present with the classic smooth brain because their single X chromosome carries the defective engine.
But females who have two X chromosomes present with a fascinating and severe phenotype called a double cortex, don't they?
They do. Due to X inactivation, roughly half of their migrating neurons have a working engine and the other half are broken.
So the healthy neurons migrate to the top, but the broken ones get stuck halfway up the trellis.
Exactly, forming a thick, rogue band of gray matter stranded deep in the white matter, which acts as a massive generator for severe seizures.
It perfectly illustrates our second rule of construction, the cascade effect. A molecular engine failure at week 10 of gestation cascades into a catastrophic structural neurological disorder at birth.
Right.
Okay, so that is radial migration. But the brain is far too complex to rely on a single travel method.
I see.
Not all neurons climb the trellis. Some take a completely different horizontal route. This brings us to tangential migration.
This represents a relatively recent, massive paradigm shift in how we understand neuroembryology.
Really?
Oh, absolutely. For decades, it was assumed that all the neurons in the cerebral cortex were born locally in the dorsal forebrain and simply climbed upward. But we now understand that a crucial population of cells, the inhibitory GABAergic interneurons, are born far, far away.
These are the cells that apply the brakes to the brain's electrical activity. Without them, the brain would just fire uncontrollably, and they don't originate in the cortical ventricular zone at all.
No, they're born deep down in the ventral forebrain, in structures called the ganglionic eminences. Specifically, the medial ganglionic eminence, or MGE, and the lateral ganglionic eminence, LGE.
Guided by an entirely different set of transcription factors such as NK by 2.1, MASH1, and DLX genes, these interneurons exit the anisole cycle and begin migrating completely sideways.
Tangentially, they travel vast distances, ignoring the radial glia entirely, to infiltrate and integrate into the developing cerebral cortex alongside the radially migrating excitatory neurons.
I picture it like two entirely different species of cells, born in completely different countries, migrating across a vast landscape to build a city together, and they have to strike an absolute mathematically perfect balance.
The balance is critical. If you do not have enough GABAergic interneurons migrating into the cortex, the brain is left with an overwhelming surplus of excitatory activity.
The clinical result being severe epilepsy.
Yes. In animal models, if you genetically knock out the transcription factors in the ganglionic eminences, say, by deleting DLX1 and DLX2, you see a 75% reduction in neocortical interneurons. The entire electrical circuit of the brain is hopelessly unbalanced.
And just to add one more layer of evolutionary complexity, while the six-layered neocortex builds itself inside out, phylogenetically older structures in the brain build themselves outside in.
Yes. The evolutionarily older parts of the brain, such as the hypothalamus and portions of the hippocampus, construct themselves in reverse.
So the firstborn cells remain on the outside, and the later-born cells must push their way into the center.
It serves as a reminder that evolution is not a master engineer starting from scratch. It is a tinkerer, cobbling together different strategies and migration patterns over millions of years to achieve a functional result.
So our neurons have proliferated, and they have migrated to their final destinations. They're sitting in their designated layers. But a brain cell in isolation is useless. To create a functioning circuit, they need to connect. They need to wire up.
Which brings us to integration, process outgrowth, and the necessity of death.
Right. Once a neuron arrives at its final location, it breaks its symmetry. It sprouts a primary axon and sends it out to find its specific target, which could be millimeters or feet away.
And leading the charge at the very tip of that advancing axon is an incredible structure called the growth cone.
The growth cone was first discovered and named over 100 years ago by the legendary neuroanatomist Santiago Ramon y Cajal. He drew them by hand, looking through early microscopes, and in his sketches, they look like little hands reaching out in the dark.
That is an incredibly accurate description. The central domain of the growth cone is packed with mitochondria for energy and microtubules for structural support.
But the outer edge, the peripheral domain, is highly dynamic and packed with actin filaments, right?
Yes, it features these veil-like extensions called lamellipodia and tiny, rapidly probing spikes called filopodia.
They function like microscopic fingers, constantly extending, retracting, and feeling their way through the dense surrounding tissue.
They are driven by the rapid cyclical polymerization and depolymerization of actin. The growth cone is constantly building and breaking down its own internal skeleton to haul itself forward.
And we know that if there are genetic abnormalities in the proteins that regulate this actin machinery proteins with names like ARP23, profilin, or cofilin, the wiring of the brain fails,
leading to severe cognitive disorders.
For instance, a mutation in a regulatory protein called limb kinase is responsible for Williams syndrome.
Yes.
This is a fascinating clinical condition characterized by profound spatial and cognitive deficits, but ironically, coupled with highly verbal, exceptionally social, and empathetic personalities.
It's remarkable. The entire structural and psychological profile of the patient is altered simply because their growth cones couldn't regulate their actin fingers properly during development.
And those filopodia fingers aren't just feeling physical obstacles, they're sniffing for chemical cues. The embryonic environment is a complex map of secreted proteins. It is a microscopic game of hot and cold.
Some of these cues are highly attractive. Molecules like netrins act as chemical beacons. The growth cone senses them and essentially says, hot, move towards that gradient.
Conversely, other molecules act as repulsive barriers.
Right, proteins like slits, which bind to roboreceptors or semaphorins and ephrins. They tell the growth cone, cold, danger, turn back.
Ephrins are particularly important for preventing developing axons from entering the wrong target fields. By carefully navigating this intricate three-dimensional map of attractants and repellents, an axon from layer V of the motor cortex knows exactly how to travel all the way down through the brainstem and into the precise segment of the spinal cord without getting lost.
It is an unfathomably complex wiring diagram, but here is the major plot twist.
Okay, lay it on me.
Here's where we return to our opening metaphor of sculpting the marble block or slashing a trail through the dense jungle. The brain does not simply build the exact connections it needs and stop.
It massively, aggressively overbuilds.
It produces an exuberant, excessive number of neurons and synaptic connections, far more than will ever be needed in the adult organism.
To sculpt the final, functional masterpiece, massive sections of this overgrowth must be deliberately and permanently chipped away. This is programmed cell death, or apoptosis.
It is a fundamental developmental necessity. Depending on the specific brain region, anywhere from 20% to an astonishing 80% of the neurons initially generated undergo what we term histogenetic cell death. They are brought into existence simply to die.
Hold on, I have to challenge that. From a purely evolutionary standpoint, expending all that metabolic energy, building the DNA, migrating the cell, growing the axon, just to kill 80% of them makes absolutely no sense. It seems incredibly wasteful.
It does seem paradoxical. Historically, it was believed to be purely a Darwinian competition for survival factors, like nerve growth factor.
So survival of the fittest neuron.
The theory was that only the neurons that successfully reached their target got the trophic nutrient, and the losers starved. While that holds true in the peripheral nervous system in the central brain, we now understand it is a sophisticated mechanism of quality control and systems matching.
So it's structural refinement.
Exactly. By overproducing neurons, the brain ensures that every possible target is heavily innervated. The subsequent wave of cell death then eliminates the errors, removes the redundant pathways, and precisely coordinates the proportions of interacting cell populations.
You overbuild the scaffolding to ensure the final building is flawless, and then you tear the scaffolding down.
That's a great way to put it.
And the mechanism of this death is remarkably elegant. It's not messy. It's not necrosis, where a damaged cell simply swells, bursts, and spills toxic, highly inflammatory contents into the surrounding brain tissue.
No, apoptosis is a highly controlled, internally managed dismantling. It is executed by a specific cascade of protease enzymes called caspases, predominantly caspase 3 and caspase 7.
So when the internal death signal is triggered during the execution phase, what happens?
These caspases activate enzymes that systematically chop up the cell's chromosomes. They meticulously disassemble the cell's internal cytoskeleton. The cell membrane then bubbles outward into neat little packages called apoptotic bodies, which are quietly and quickly swallowed up by neighboring immune cells called phagocytes.
It leaves zero trace and triggers absolutely no inflammatory response. It's like biological Marie Kondo. Thank the cell for its service and neatly discard it.
Exactly. But what happens if this crucial pruning process goes wrong?
Well, if you genetically delete those caspase enzymes in embryonic mice, their brains become massively overgrown, chaotic tangles.
Which brings us back to clinical psychiatry. We are increasingly viewing several major psychiatric and developmental disorders as fundamentally being diseases of faulty pruning.
In autism spectrum disorders, for instance, structural MRI studies often reveal a period of early brain overgrowth in the first few years of life.
A prevailing theory is that this is driven by insufficient early pruning. Too many synapses and connections are left intact. The jungle is too thick.
This hyperconnectivity can lead directly to the sensory overload, noise, and processing differences characteristic of autism.
And on the flip side, consider schizophrenia. It classically manifests in late adolescence or early adulthood.
And this timing isn't random. It perfectly coincides with the final major waves of cortical pruning sweeping through the prefrontal cortex.
Exactly. The dominant neurodevelopmental model for schizophrenia suggests an excessive runaway pruning process during this critical adolescent window.
The brain chips away too much of the marble, slashing too much of the jungle trail.
This leads to a measurable loss of cortical gray matter, which correlates clinically with the disorganized thoughts, negative symptoms, and onset of psychosis.
Too little pruning, you get overwhelmed by noise. Too much pruning, you lose the signal entirely. It beautifully synthesizes the biology and the clinical presentation. A psychiatric illness isn't necessarily an acute infection or a sudden break. It is a developmental sculpture that has either been under-refined or overcarved.
Spot on.
This brings us to our final major section, late vulnerabilities in adult neurogenesis. Everything we have discussed so far happens primarily in the womb or very early infancy.
But neurodevelopment doesn't strictly stop at birth. Certain populations of neurons have drastically extended windows of generation.
And the prime example of this extended vulnerability is the cerebellum, the little brain tucked at the back of the skull.
It is densely packed, containing roughly 75% of all the neurons in the entire human brain.
Yes, and the vast majority of those are tiny cells called granule neurons. While the large, complex Purkinje cells of the cerebellum are born quite early in gestation, the granule neurons are generated from a secondary proliferative zone known as the external germinal layer, or EGL.
And crucially, the EGL sits right on the outermost surface of the cerebellum. But here is the wild part. In humans, this superficial layer keeps actively churning out new granule neurons for up to 2 years after birth.
This extended window carries massive clinical meaning. Because this massive stem cell population is sitting right on the surface of the brain, actively dividing throughout the first 2 years of a toddler's life, it is uniquely, profoundly vulnerable.
It is vulnerable to postnatal viral infections. It is vulnerable to early childhood chemotherapy.
And historically, it was highly vulnerable to standard medical interventions, wasn't it?
It was. It used to be common practice to administer high-dose corticosteroids in the NICU to premature infants to help mature their lungs rapidly.
But steroids inherently inhibit cellular proliferation.
Right. Clinicians eventually realized that these early life-saving steroid treatments were inadvertently halting the proliferation of the EGL. They were permanently damaging cerebellar neurogenesis, leading to long-term cognitive and motor delays in those children.
It highlights the delicate balance in neonatal medicine. And the vulnerability of the EGL cuts both ways. If it stops proliferating prematurely, you get developmental delays. But what if it loses control of its proliferation? What if the molecular stop signal breaks entirely?
You get a tumor, specifically medulloblastoma. It is one of the most common and most devastating solid childhood brain tumors.
Medulloblastoma is essentially a cancer of the external germinal layer.
It is, driven by mutations in the very developmental signaling pathways we discussed earlier, like sonic hedgehog and Wnt. The tumor is simply a developmental process that refuses to turn off.
Okay, so the cerebellum finishes its major neurogenesis by age 2. But that raises the ultimate question. The question that has caused more arguments at neuroscience conferences than almost any other topic.
Oh, I know where you're going with this.
Are we stuck with the neurons we have at birth or shortly thereafter? Can the adult human brain grow new neurons?
Adult neurogenesis.
Yeah.
It remains perhaps the most fiercely debated topic in modern neurobiology.
So what's the verdict?
Well, in rodents and non-human primates, it is definitively, indisputably proven. It is highly restricted to 2 specific zones: the subventricular zone lining the lateral ventricles, and a tiny sliver of the hippocampus called the subgranular zone of the dentate gyrus.
And in rodents, we know these newly born adult neurons are fully functional. They integrate into existing neural circuits. They're absolutely required for forming specific types of trace memories.
If a mouse suffers an ischemic stroke, the SVZ will actually pump out new neurons that migrate directly to the site of the injury in an attempt to repair it.
That's incredible. The mechanism is clear in lower mammals. But humans are vastly trickier to study.
Very tricky. The textbook highlights this ongoing controversy. Some prominent postmortem studies of healthy human brains look for a specific protein marker called double cortin, or DCX, which we mentioned earlier as the microtubule engine required for migration.
Right. DCX is a highly reliable marker for immature, newly born neurons. Some leading laboratories have found thousands of these DCX-positive cells in the adult human hippocampus, even in individuals in their 70s and 80s.
However, other equally respected laboratories, using very similar staining techniques, report that this neurogenesis drops to virtually undetectable levels by the time a human reaches age 18.
Why the massive discrepancy? How can 2 labs look at the human brain and see entirely different things?
It largely comes down to the extreme fragility of the tissue and the history of the patient.
Okay.
The labs that successfully find the newborn cells note that the DCX marker degrades incredibly rapidly if the postmortem brain tissue sits in a fixative solution for too long, which is standard practice in many brain banks.
And the labs that fail to find the cells have historically relied on tissue banks that extensively fix the brains.
Furthermore, early studies often use tissue from patients with a history of severe epilepsy, a condition that might have physically destroyed the delicate neurogenic niche long before the patient died.
So while the jury is still technically out, many leading neuroscientists strongly suspect that human adult hippocampal neurogenesis is real and vital, even if it operates at a lower volume than in a mouse. Let's assume it does play a significant role in humans, because the psychiatric implications are just too profound to ignore. How does our lived experience, our environment, physically shape this birth of new neurons?
It acts as a biological barometer for stress and enrichment. The negative modulators are intensely powerful. Severe psychological stress, chronic social isolation, and specifically sustained high levels of glucocorticoids like the stress hormone cortisol strongly completely inhibit neurogenesis in the hippocampus.
Cortisol essentially shuts down the factory floor.
It does, and this provides a direct biological explanation for a phenomenon psychiatrists have observed on MRI scans for years. Patients suffering from severe chronic major depression, PTSD, or chronic schizophrenia consistently show measurably reduced hippocampal volumes.
The relentless stress and elevated cortisol have literally halted the maintenance and growth of their memory and emotional regulation centers.
But there is a hopeful, powerful inverse to this: the positive modulators. Environments rich in stimulation, consistent physical exercise. They actively stimulate neurogenesis.
And remarkably, so do antidepressant medications.
Yes.
This is the absolute clincher for me. SSRIs, like fluoxetine, don't just tweak the levels of floating serotonin in the synapse to make you feel temporarily better.
No, studies in animal models and correlating structural MRI data in humans demonstrate that chronic administration of antidepressants actually stimulates the progenitor stem cells in the hippocampus to multiply, survive, and mature into new neurons.
It completely reframes how we conceptualize psychiatric treatment. Treating depression might not just be about altering a transient chemical imbalance.
Yeah.
It might literally be about fertilizing the brain, aggressively growing new neural infrastructure to rebuild what was withered away by chronic stress.
Growing new neural infrastructure. That perfectly bridges the gap between the purely biological brain and our lived experience.
All right, we have covered an immense amount of ground today, from the primitives tree to adult neurogenesis. Before we wrap up, for those of you preparing for board exams or just wanting to solidify your clinical reasoning, let's run through a few clinical scenarios you might actually see on the wards, drawn directly from Chapter 1.4 of Kaplan and Sadok.
Sounds good.
I'll present the clinical concept, pause for a moment so you can think it through, and the expert will walk us through the answer and the rationale. Ready?
Ready.
Scenario 1. A fundamental concept. When looking at the complex, six-layered architecture of the mammalian cerebral cortex, what is the primary mode of cellular generation and migration that establishes this structure? Pause for a second to think.
Right. The answer is inside to outside radial migration. Remember the trellis analogy. The deep layers, V and risers, are established first at the bottom. The later-born neurons must physically migrate radially past those early cells to form the superficial layers 2 through 4.
Perfect. Scenario 2. A clinical mechanism. You are consulting on a fetus diagnosed via ultrasound with a severe neural tube defect resulting in spina bifida, a deficiency in which specific maternal dietary supplement or exposure to which common psychiatric medication is most classically associated with this failure of neurulation.
Well, the answer is folic acid deficiency or valproic acid exposure. These agents disrupt the precise chemical gradients and cellular machinery required for the neural folds to elevate and zip shut during month 3 and 4 of gestation.
Scenario 3. Clinical reasoning in the NICU. Premature infants treated with high-dose corticosteroids to mature their lungs are known to be at risk for subsequent motor and cognitive delays. Which specific brain structure is most vulnerable to this postnatal insult due to its prolonged period of neurogenesis, which extends up to 2 years of age?
The answer is the cerebellum, specifically the external germinal layer, or EGL. Because this superficial layer continues to actively divide and produce granule neurons long after birth, it is uniquely susceptible to anti-proliferative drugs like high-dose steroids.
Scenario 4. An embryological mechanism. We know the cortex relies on inhibitory GABAergic interneurons to prevent runaway excitation. However, these cells do not originate in the cortical ventricular zone. Where do they originate, and how do they reach their final destination in the cortex?
They originate deep in the ventral forebrain, specifically in the ganglionic eminences, the MGE and LGE. They reach the cortex via tangential migration, moving completely horizontally across the developing brain rather than climbing radially.
Scenario 5. Another clinical concept. You are reviewing the genetic panel of a patient with profound intellectual disability and a completely smooth brain surface on MRI. A mutation of the gene encoding the glycoprotein reelin is most likely to result in the severe structural abnormality. Why?
The resulting condition is lissencephaly. Reelin acts as the molecular stop sign secreted by Cajal-Retzius cells at the top of the cortex. Without it, migrating neurons fail to detach from the radial glia and organize properly, destroying the inside-out lamination and entirely preventing the cortex from folding.
And our final scenario, scenario 6. During normal, healthy neurodevelopment, what is the primary intended consequence of activating the intrinsic apoptotic pathway via caspase-3 and caspase-7?
The answer is histogenetic programmed cell death. This is the crucial pruning mechanism. It safely and cleanly dismantles 20% to 80% of excess, overproduced neurons without triggering any toxic inflammatory response, refining the neural circuits for optimal efficiency.
Fantastic. If any of those concepts felt murky, I highly recommend diving back into Chapter 1.4 in Kaplan and Sadok's comprehensive textbook of psychiatry. Before we close, I want to leave you with one final provocative thought to mull over.
Oh, excellent.
We just discussed how chronic stress and high cortisol shut down the birth of new neurons in the adult hippocampus, while environmental enrichment, learning, and antidepressants actively promote it. Our lived experience literally changes our brain structure. But think back to the subplate, that temporary fetal waiting room we discussed. Those early neurons are wiring up the brain, firing spontaneously, physically practicing the connections before the baby's senses even turn on. If the brain is physically practicing its wiring in the dark of the womb, shaped only by early genetic cascades and the maternal chemical environment, how much of our baseline resilience to future psychiatric illness or our vulnerability to stress is already structurally sculpted into the marble before we take our very first breath? Here are the details of this episode.
Neural development and neurogenesis. Byline. From formation to refinement, how the brain grows, shapes itself, and learns what to keep.